Yeah. Hello, everyone, and welcome to yet another Journal Club brought to you by Leaf and Lifespan.io. I'm Dr. Alvin Medvedik, live streaming from my lab, the Canberra Center of Biomedical Engineering here at the Cooper Union, the Lower East Side of Manhattan. And today's paper is going to be another back-to-back paper following the last telomere paper. So this paper, we're going to focus on a very specific activity that telomerase possesses, a pause function. And we'll talk a little bit about the implications regarding what this might mean when it comes to therapeutics. So without much further ado, let me share my screen and go right to the paper. All right, how's that looking? Does that look good there, Steve?
It does, yeah. Looks fine by me. Nice and readable. And that's pretty good. It must be because I'm short-sighted. So, yeah.
Excellent. Okay, so the article is published very recent, February 12, 2018. A single nucleotide incorporation step limits human telomerase repeat addition activity by Chen et al. From, I believe, somewhere in Arizona State University, Tempe, Arizona. States. So what is this paper all about? So unlike a lot of the other papers we've been looking at, which basically are cell biology related or dealing with mouse models, this paper here is a pure biochemistry paper. They wanted to essentially look at a particular function of telomerase. Telomerase is the enzyme that most of us are aware adds repeats of DNA to the ends of linear chromosomes. And it's a reverse transcriptase, meaning that it uses, it basically converts a sequence found on RNA, which it carries around as a template. So it's really, telomerase is a ribonucleoprotein. And it uses that to synthesize repeats of DNA at the ends of chromosomes because of a problem known as lagging strand. Well, it's not a problem, but due to lagging strand synthesis, every time cells replicate, they lose a little bit of themselves. And human telomerase synthesizes repeats that come in the form of GGTTAG and the little sub-N, which means they're just repeated over and over again. And these repeats are specific to different species. So depending on which template this telomerase carries, you get different types of repeats. So what are these guys really concerned about? Well, they wanted to study the rate limiting step basically for telomerase addition. So telomerase as an enzyme, as a polymerase, let me just, stop sharing. Oh, there we go, back to my face. Telomerase as an enzyme has several functions. It's there to essentially replicate DNA. It's there to extend DNA by adding nucleotides to the 3' end. And so there are really kind of a couple of functions that need to be realized. One is processivity. So once the enzyme starts adding repeats, how long does it keep adding repeats, right? Until it falls off because it's adding them in a linear manner. The other aspect to this enzymatic function is how fast does the enzyme go, right? So how fast are those repeats added? How fast are those nucleotides incorporated? And a third aspect to this enzyme that's particular to telomerase is shifting of the template. So after it basically synthesizes 6 of these repeats, it then needs to move the template sort of like a typewriter a little bit, over by 6 until it could then synthesize 6 more. So the question is, is there anything rate limiting here for telomerase activity? And it turns out that there is a crucial rate limiting step, and it is the incorporation of the first nucleotide, which would be GGTH, which would be the first G. And as opposed to all of the other functions, this incorporation of this first G, although the authors then go on to state it doesn't have to be a G, it could be some. If you change it artificially, it could be an A. You still get the same pause. So there's this pause function, right? So the authors then do a number of biochemical assays to basically peel apart how this pause function functions. All right. So let's go back to the paper. Okay. So there's a lot of terminology here. There's a lot of jargon, as all papers have jargon. This one has its own particular jargon. So the authors had to, this is all of these figures here in figure 1, these subfigures, this is all done in vitro. So it's a cell-free extract. In order for them to see how this enzyme is functioning as regarding rate, so I'm not going to go too much into enzymatic measurements, but what they are doing here is calculating something known as the Km value. And that's really, so I mentioned earlier that as an enzyme functions, there's a speed to it, which at least as far as the polymerase is concerned, adds nucleotides, right? So at a certain concentration, given a certain concentration, there is a maximal velocity by which this enzyme can function, right? It can't function at infinite velocity. There's a certain speed at which it can add nucleotides. And half of that speed, half the Vmax, is known as the Km value, right? So basically what these numbers here, and I'm just going to quickly dive here and just briefly mention this because they talk about this a lot in the paper. This 120 plus minus 8 micromolar, 400 plus minus 8 micromolar, that's basically the concentration of this particular substrate, which is a nucleotide that's required to give you, well, that's the Vmax. So at this concentration, this is the half velocity of the enzyme. So basically what this means is you need more of a particular substrate to reach half maximal velocity, which means that the higher the Km value, the slower the enzyme, right? So that's the slower the step. So the lower the Km, faster it is, right? So I guess you want to consider that to be a better enzyme, then the lower the Km value in these cases, the better it is, the faster it is incorporating nucleotides. So in order to kind of measure this incorporation and see, within a given unit of time, how much is incorporated so they can make these calculations, they had to basically devise an artificial system where they have TERT, the catalytic subunit, which is the protein subunit that was synthesized in cells, assembled with these artificial bits of telomerase RNA, HTR, telomerase RNA, PK, which is, I believe, abbreviated to pseudonot, and this other one, CR45. These are required basically for like the structural stability of TERT and how it basically can line up with this template, which is this green part here. And into this mix, they throw in a DNA primer. So you can see that, this template is a very short piece here. And what it enables the researchers to do is basically, this telomerase can't, this telomerase is only able to extend on this defined length of primer. So you don't get a lot of, crazy sizes. So basically that's how they came up with this assay. And over here in C, so they don't show the data here. It's in the supplemental and other figures. But what they discovered was, and we'll go into this figure over here, is that prior to the first letter that's incorporated, so I said it's GG, TT, AG, these numbers 1, 2, 3, 4, 5, 6 refer to the numbers that are coming right at the edge of this, where the DNA is, the last nucleotide incorporated. The first one that is normally there is G1. And in order for it to pause here, it requires this TA sequence, which is referred to as a pause signal. And they mentioned also the other A is important as well. So what this means in practice is that if you look at the Km values, this is wild type. Delta G1 is 120 micromolar. And then, well, they don't have 2 here, but T3 is 4, A5 is 31, G6 is 5. So essentially, this is a slow step. And regardless if you add in another nucleotide, which they do later, if you change this template to incorporate an A or a T, this very first nucleotide incorporation is a rate limiting step, right? So this is where it pauses, right? So essentially at the beginning of synthesis is where this telomerase pauses. So they were able to do a number of assays here. They were able to show that you can essentially overcome this pause by increasing the concentrations of the first nucleotide incorporated. In this case, this was deoxy-GTP. These templates were radio-labeled, so P32DATP, so you can see them on a gel. This is a loading control up here. And you can see that, so these are the numbers G1, G2, this is G6, so this is A5, that's the radio-labeled one. And you can see that as the concentration of GTP is added, you start getting more and more incorporation of, well, you get a longer piece of DNA that has the delta G1 on it, essentially. So this is just band intensity that they measure, right? So, so that's really the take-home message of that figure here. Stop sharing the screen here. Okay, move on to the next one. So this paper is basically, you know, a lot of gel analysis of telomere addition, right? So, any questions?
Nothing yet, nothing so far.
Okay. All right. So it's a very good meat and potatoes paper. I don't mean that derisively. I mean that in a good way. The same way that the first CRISPR paper was a very meat and potatoes paper where Doudna and partners really did a lot of biochemistry to show exactly how the enzyme functioned, the Cas9 enzyme in vitro. They're going all out to figure out exactly how human telomerase functions in vitro. So this pause function basically does, you know, here it's just a little figure showing that this pause signal does two things. It defines the 5' boundary, right? So basically it pauses whatever is there, right? It doesn't have to be a G. And it also, and in the wild type case, it limits the first G incorporation, right? So that's what it normally does. And I've got to refresh myself by staring at these images here. Which assay is this? Direct primer extension assays. Okay. High molecular weight, low molecular weight. This is P32 labeled substrate. Increase of ATP doesn't do it, but in this assay here, what they're doing, this array is they have a substrate that's radio labeled, and all of this basically this pattern here that you see is longer and longer telomeres, right? So these are P32 labeled. So the more additions keep getting added, and when you run it on a gel, the higher these blotches become up here. It's higher molecular weight, low molecular weight. So that's an acrylamide gel. So adding in more GTP, what you notice is that you overcome this pause and you get more extension, right? What are they doing on C here? Let's see, P32. Is that a different substrate or not? There's a lot of stuff here. Well, I think I'll go back to this because I'm not gonna pause here to read the figure legend. Figure 3. Okay, so this is where they start to characterize the other two functions I mentioned about telomerase, which is the processivity and the rate, right? So how far does the enzyme go before it falls off, and how fast does it go? And they analyzed the processivity using this assay with the substrate on beads, or actually it may have been the enzyme that was bound to beads. And it was basically a pull-down assay, right? So essentially, processivity is a function of how far the enzyme is going. So that means if the enzyme is still going, it's still attached to the substrate. So if you pull down the substrate and it's attached to the enzyme and it comes down, then you get a lot of bound material, right, versus less bound material, which means the enzyme is falling off at an earlier stage. So they were able to calculate the relative processivity, and this is bound versus released, so it would be bound to these beads. So with an increase of delta GTP, which is the wild type, you get more processivity, of course. And the rate was determined, and I'm not going over these figures, I'm skipping them because this is the quantification of what you see here, the relative fold change that you see in processivity. The figures below are a rate analysis, and the way they're doing it here is basically what's called a pulse chase. So what they do is they add in radio-labeled nucleotides, in this case deoxy-T and T, and after they pulse it for 5 minutes, meaning you have 5 minutes of extension happening, you then chase it with a cold nucleotide, and these are 10 micromolar or 100 micromolar DGTP, and these are 5 minutes, 10 minutes, 15 minutes, 20 minutes, right? So if the enzyme is going faster, what you would expect to see is as you increase the concentration, you would expect to see this angle basically be much sharper than this angle here, which it is, right? So you see much higher molecular weight within the same given unit time, right? So 20 minutes here, 20 minutes here, you have a high molecular weight. And adding in other nucleotides, in this case DATP, is not, and this is basically their extension rate calculated, extrapolated from this data here, 0.86 to 1.45, so it's about a twofold extension, a little bit more than 2.5-fold extension, and we don't see any difference when you chase it with high concentration DATP and let it go for a longer period of time. So basically what the authors are saying from this is that that both the processivity and the rate of incorporation, so you can see here when you put, you have much more processivity occurring when you add in a lot of GTP here, meaning that, the enzyme is stuck to the template much longer. Both of them are influenced by this pause function, right? So essentially that first nucleotide is required, it's both rate limiting for the speed at which the enzyme is functioning and also how long the enzyme is attached to the substrate. So that first nucleotide, and also it's the, and the, I guess the other nucleotides required for the pause function, both of those are required for this effect, right? Okay, back to sharing screen. This figure here is, I mentioned earlier that it's independent, this pause function, of which particular nucleotide you have. So they changed the G. So these are mutant versions of the template. So delta G, deoxy G1 is changed to deoxy A1, right? And also this C here, or deoxy G2 is changed to A2. So meaning for the RNA version, right, the A is base pairing with a U, so you have this uracil instead of a C, right? So just remember, this is the template, and this is the DNA that's being incorporated, right? So you have your reverse complement here with your uracil that's replacing your T. So they made these various mutant templates, and they showed, the take-home message basically is that this is wild type. You have increasing GTP here, ATP, 51U, so we're going to focus here. This is the rate limiting one. And right here, what's happening is it's not going up with the addition of more GTP, but it is going up the rate, the incorporation or the length of telomeres with the addition of extra ATP. Right. So I'm not going to slog through the rest of it, but essentially it's independent of the nucleotide. And this is basically the relative ratio of the intensities for this figure. So it's nucleotide independent. Figure 5. Yes. So now, just like they did for the earlier figure, they want to check the effects of DATP on repeat addition processivity, and the rate for telomerase mutant HTR51U. So, which one is that? That is, yeah, that's the one with the first nucleotide that's changed. And just like what we've seen before, having more DATP added increases the processivity. It also increases the extension. So we go here from, so this angle here, so this is again a pulse chase experiment, but in this case with DATP. So basically this is reversed, right? So here we have DGTP, right? So this is a GTP that's downstream. That's not changed, right? So we have 2.9 to 2.95, and here we have an extension rate of 1.27, go to 3.15. So that's about, again, a twofold increase in rate of addition of nucleotides just by switching from a G to an A. So it's a completely different nucleotide. A's have only 2 base pairs versus 3 base pairs. So that basically hones in on this, whatever that N1 nucleotide is right after the template, that's going to be where the enzyme stalls, right? So that's going to be essentially important at stall. So, any questions, Steve?
I always forget where the unmute button is on this thing. It's like, we do have a question. Yuri has asked, how can we make sure that only good cells are affected by telomerase extension therapies? Is there a good way of targeting at this level? And do you personally think that this type of therapy has a chance of becoming one of the first available to the public? Well, there's actually really 3 questions there.
Yeah, there's a lot in there. Well, the question, the implication of that question, and I'm just going to answer part of it, Steve, you can flip the rest, but it implies that, we are able to come up with a therapeutic that's going to alter the rate of incorporation and the processivity. This paper, there's only one sentence at the very end of the paper that says, this suggests a therapeutic intervention. But really the bulk of the paper, besides that one sentence, is teasing apart the mechanism. So clearly flooding the cells, with extra, whatever the first nucleotide is, GTP is not going to do the trick. That's not practical or feasible. So you want some way for the first nucleotide to be incorporated, quickly. And one way to do that, most practical way, and they talk about it later in the paper and those figures, is to, somehow mess around with the pause function, which is the nucleotides that are within the template, but further away from where that first nucleotide incorporation site is. The question is, how do you do that, right? You need to have some sort of molecule that would be specific enough to bind to that binding pocket for the template, and also not screw up the shifting of the template, as it's working. So this paper really doesn't go into how such a therapeutic would, be able to do that. It suggests, the only thing the paper really suggests is that this is probably the area of the enzyme that you would have to go after somehow to basically, weaken, that, those A's at that location, within the template. Now, what sort of molecule you would use and how you would use it, that's your guess is as good as mine.
Early days, really, isn't it? So
yeah.
So do we think that this type of telomerase boosting therapy to increase the length of telomeres is likely to become one of the first available? Personally, I would say no. I think it's up there along with epigenetic partial reprogramming, like we talked about last year with Belmont. I mean, if it can be made to work, I think it's going to be more one of the more technically challenging. So I think we're a good, probably 10 years or so out before that sort of thing gets deployed. And
10 years is a stock answer that I also give for I don't know. 10 years.
Well, yeah. I mean, I think we'll see clinical trials within possibly the next 5 years. The way that people like Blasco and Ajax and people are pushing, it could be sooner rather than later. But mainstream adoption of things like that, providing they can work, got to be about 10 years or so.
Yeah. So, I mean, clearly, well, depending on who you talk to, right? I mean, clearly, I'm not going to go too far in this direction, but Steve knows what I'm talking about. Elizabeth Blackburn's work shows that, telomere lengths are, rather flexible, alterable in adult cells through a variety of different interventions. So, a pharmaceutical intervention that could do that as well. Sure, why not? I just, again, based on this paper alone, it doesn't give us it gives us a suggestion for what would be needed to speed up the activity of telomerase. But remember, the telomerase and its RNA component have to be in the cell, right? So you have to have hTERT expressed in the cells to begin with. You have to have your RNA expressed to begin with for any small molecule that would accelerate the activity of telomerase. So I don't know if this telomerase is still floating around in an inactive form, but if it's in an inactive form or it's not expressed, then a molecule that would bind to that site and somehow undo the pause function wouldn't really be applicable, since the telomerase is either not there or bound up with something else. So it's really would only apply for cells that are already active producing telomerase, and whether or not we would need to accelerate telomerase in that case is also a big question. I mean, would you need to accelerate telomerase activity if it's already expressing telomerase? So that's a question, that's not answered right now.
Well, there is some data to suggest that even our stem cells, lose telomere length. So obviously, it's not keeping pace with the with the repeats. That's what you really want anyway. I was going to mention that you don't want to hit somatic cells and give them telomerase anyway. It's a bad idea because one school of thought suggests that giving aged and potentially already damaged cells additional replications or indefinite amounts of replications would be a bad idea. Now, the stem cells, obviously being a little bit more robust and about repairing themselves, are the ideal sort of target. In fact, they even talk about that in this paper. The fact that, if we could get that kind of selectivity, then, and we can only treat stem cells, then, that has potential. But I think that really applies to the majority of telomerase therapies, whether it's viral vectors or small molecules or even a system that targets this pause function. I think that broadly applies to all of those.
Yeah. So great question, but there's a lot of moving parts in that question.
More research.
Not easy to answer.
More research needed, right?
Yeah.
But yeah, interesting.
So speaking of the pause function, I'm skipping figure 6. And I'll tell you why I'm skipping figure 6, because this figure talks about how similar human telomerase is to other polymerases in that it also can utilize guanosine diphosphate as well as guanosine triphosphate, and that's great, but I'm not sure it's very relevant to what we're interested in. So they compare it to Taq polymerase, Klenow fragment of DNA polymerase 1, an intron reverse transcriptase, and GMP doesn't work, which is guanosine monophosphate. GDP does work if you add higher concentrations. So the reason I wasn't really 100% sure why I did these experiments. I did when I read the paper, but I'm just going to put that on the back burner here. So it functions like a lot of other polymerases do. Okay. So kind of the figures that are most interesting, I think, core figures for for us in the paper are figure 7 and figure 8, which is what happens when you get rid of the pause signal, right? So pause site, the pause signal, dictates where the pause site is, and it's this A, right? So this is not the first nucleotide incorporated; it's over here. So what they did was they synthesized something that has these pause signals changed. So they changed all of these A's to U's, basically reversed it, switched it around. So where there was an A here, they converted to U. Where there was a T here, they converted to an A, right? A's and U's and A's and T's basically go to peanut butter because of base pairing rules. And what happened? This is their in vitro assay that they worked out earlier. And the intensity of these bands basically tells you how much telomeric DNA there is, and increasing amounts of DGDP, which is the first nucleotide incorporated. And you could see, within the same given unit of time, a lot of extended telomeres, right? So this enzyme seems to function a lot better, and they were able to calculate I believe the Km value. I don't know where it is though. It's a lot lower though. And the follow-up experiment is, again, seeing what happens. Does getting rid of this pause function, how does it affect the processivity and the rate, right? And I'm going to first, where's the rate? Rate. Where's the extension?
So you said you mentioned how to do a temperature gradient.
Yeah. I'm going to go back here. So this is the processivity. What's interesting though is, so this is wild type. When you add more DGDP, obviously you have more processivity. This is bound with delta pause. Again, it seems to be a lot from the beginning. It doesn't matter if you have more DGDP. It seems to me, and they may briefly mention it up in the paper, I'm not sure exactly where, but you do tend to have a missing amount of very high molecular weight telomere ends. So, I mean, that to me, again, I may be misinterpreting it, but it's suggesting to me that perhaps the processivity is affected, right? So if the rate is going up and you're synthesizing like crazy, maybe it's also prone to falling off. As well, even though they're still relatively high, but I don't see species that are as high with the wild type, right? Though you have a lot more. So I know the rate was increased. Just trying to find the rate values here, because right here what they're doing is they're increasing the concentrations of DGDP, and oh yeah, I know why this confused me, because they compare it to figure 3, which was kind of a little irritating to me. So the extension rate here is 1.7 and 1.63, and it doesn't go up. So with adding more GTP, suggesting it's already running at a high rate. So obviously the question is, well, that's the mutant with the delta pause. Where's the wild type without the delta pause, the rate? And that's the experiment that was done back here. So, here, yeah. Here. 1.45. I think it was 3. 3.45. It was an earlier figure, and I have to find it in text where they referenced it. Oops. So let me see. extension rate, 1.7, 1.63, plus or minus 0.007. That doesn't appear to be that much higher. Let me find the sentence in the figure. Crucial determinant sensitivity. Yeah, figure 3. Pause signal. Pause. Here we go. Delta pause mutant had an approximately one-fold greater processivity at under 10 or 100 micromolar compared to wild type enzyme. Repeat addition rate of delta pause mutant is found to be 1.7 repeats per minute at 10 micromolar and 1.65, which is significantly higher than the 0.7 and 1.3 repeats per minute of the wild type enzyme at 10 and 100. Okay, so yeah, figure 3D and E. So 0.7 and 1.3 at 10 micromolar and 100 micromolar GDP versus 1.7 repeats per minute and 1.65 at 10 and 100 respectively DGDP. So it's about a 1x increase. So the enzyme is going twice as fast, right? They say that the pattern of the processivity is looked a little. Way back here. Looked a little different. Again, I might be misinterpreting this, but it seems to me that it may be not as processive just because you lack a lot of higher molecular weight, that are as high as the wild type with the delta pause. It seems to make sense to me if the enzyme's going faster, maybe it's more prone to falling off. So what would that entail? What would that mean for a therapeutic? If you get it to go faster, but the telomeres are not as long. Again, question. All right. So that takes us to the figure where they have the working model of the telomerase catalytic cycle, right? And basically what this is just in this rather complex looking figure, although it's not that complicated, they just tell you where the slow step is. So based on a lot of other people's work and their past work and the work they did here, really it's this pause function. It's this, whatever's here, this A, will dictate how quickly a nucleotide is added right at the first position, right? So once that is completed, the template translocates. And the template translocation, they mentioned somewhere in the paper, is like 100 times faster than incorporation. So it's not this template shifting over to the next 6 letters is fast. Once it translocates, then you have the slow step. So now you have the delta G1 is incorporating here very slowly. But once that's incorporated, the next series of nucleotides go rather fast, right? And the translocation to the next one. So it's basically like a pause function. That's basically what they say, right? So it's the enzyme pauses right at the first step, and there's this pause signal which is mediating this pause. So like I said, that's fundamentally the paper. It really drilled down deep, but there's still lots of questions out there. Precisely how is this pause function working, right? I mean, you have a ribonucleoprotein that it's complex with telomerase, it's complex with DNA. So how is this? How does the adenine or the uracil at that position affect the incorporation of the first incoming nucleotide, right? It is affecting it, but exactly how at the molecular level, which bonds are being affected, they don't really mention in the paper. So there's a lot of, there's still a lot of very detailed work that needs to be done before, you could use this to rationally design a drug. You could screen for drugs and then, crystallize them with the drug bound and see where it binds. And it'd be interesting to see if a drug that increases the rate of telomere repeat addition binds into that area where that pause function is. So that would be, that would then give credence to, the belief that we can rationally design a drug that can speed up the activity of telomerase, assuming telomerase was already in the cells to begin with. Otherwise, having such a drug would work. So we really drill down deep into telomerase in this paper. I think this is probably somebody's. So I'm in a lab. That whacking you hear in the background, I believe somebody is trypsinizing cells, trying to release them from the bottom of the petri dish. So it is a working lab that we're live streaming from. So those are the noises.
This is the sound of science. But there you go.
Yeah.
It's either that or you've got roaches in the building, right?
Well, I haven't seen a roach in this building, but I'm sure they're out and about.
It's New York. Of course you've got roaches. I've seen the film Joe's Apartment. Good film, actually. I don't know if you were. Yeah, it's about cockroaches. It's one of them CGI films. It's good. It's good fun. It's not what you think. The cockroaches are the good guys. So yeah, we've drilled down into telomerase there. The question is, can we exploit that mechanism? Can we supercharge, as they suggest, our stem cells? What do you think?
And then go. So you can set up.
I guess it's too easy for me to say. I don't know.
More research needed.
Yeah, always more research. Research is never ending. Of course, that I'm not. That's not to be pessimistic. We have therapeutic interventions that we can apply before we know 100% of what's happening. We just need to know enough, right, to start applying therapeutic interventions. So all in all, it seems that having longer telomeres to begin with is a good thing, right? So how we achieve that, for those of us who have shorter telomeres in our stem cells, remains to be seen. What interventions work the best?
Yeah, and epigenetics and telomere biology seem to be intrinsically tied together, and the weight of evidence suggests that epigenetics is the more important of the two, because whenever you alter epigenetic changes such as methylation, it tends to pull the telomeres along with it anyway. So I'm sort of coming around, Oliver, I don't know about yourself, that we might not even need ultimately to do anything about the telomeres. I think they will just, I think they'll just follow along, maybe. Like we saw with the OSKM program in Glasgow, that Glasgow did. It does appear to suggest, the literature does, that that may be the case. So we might not need to worry about telomeres unless in the instance of severe erosion for example in certain diseases. If we're very, very lucky, it may be a question of just resetting the epigenetic hallmarks, and it resets the telomeres to boot. It does in mice.
Yeah, I think eventually, resetting the telomeres is a necessity, clearly, because you need to, what gets you first, which damage is the most, so in the hallmarks of aging, you got all sorts of different damage that accrues, and which damage gets you first, it might even depend on the individual person. For some people, it's beta amyloid plaques, not telomeres per se. It might be telomeres for other tissues.
Well, I mean, given I had a talk with Steve Horvath over at Undoing Aging that we've just got back from in Berlin.
That's right.
And the data he was presenting and the conversation, it does strongly suggest that epigenetics really is sort of like the main, I wouldn't say it's the cause of aging, but a cause of aging. He does study telomere biology himself. He's very interested in telomeres, but he does seem to think they're kind of a second fiddle, really. I think they're important in aging, personally, but I think ultimately epigenetics and changes to your DNA are probably the two things that drive aging as a primary process.
Well, it's certainly the case with yeast cells. When you do lifespan analyses, a big bit of lifespan of yeast is not affected by telomere length. Their telomerase is upregulated. It's all the other junk that accumulates in the other cell.
Yeah, and I mean another example is the Horvath clock, and how accurate it is when measuring biological age in comparison to, say, using telomeres as a measurement, which are, you know, pretty hit and miss, to be honest, depending on what tissue you look at. They're not really a very ideal biomarker, which again strongly suggests that epigenetics is really sort of the sort of primary there, because, you know, when you look at epigenetic aging, like the methylation clock, for example, it is a much stronger correlation between it and actual biological age than telomeres. You know, telomere length is and biological age, and I mean by a massive sort of margin. It's so much more accurate, which again, it suggests that, you know, the telomeres are important, but they are a second fiddle.
Well, it would be important, you know, for dividing cells, but the bulk of our cells obviously are in a non-dividing state, right? Like those ages work. So epigenetics will definitely might play a greater role, certainly in the bulk of our cells. Perhaps we should do an epigenetics paper, maybe something related to the Horvath clock, or maybe a review, you know, regarding epigenetics in general. I don't know.
Well, I mean, seeing as we've been speaking to Steve Horvath, and you know, we have got a translated interview coming up in the week, which dives quite deep into the epigenetic clock, or should I say clocks, because as you recall from his lecture in Berlin, he's now talking in terms of clocks because there are a number of them. Methylation is only one of them. Perhaps we should take a dive into some of his more recent research and, you know, see if we can sort of come to a conclusion. But yeah, I mean, certainly biomarkers are a pretty interesting topic in of themselves, really. So yeah, we could do that, I think. The word is still out. The jury's still out on telomeres. We really, I don't know. We really don't know.
Well, they're clearly important for maintaining generations, right? So without telomere, without telomerase, and, you know, your species will not. So we're not saying they're not important; they are crucial.
Yeah. And I mean, another application they could potentially be useful for, and why we need to develop these therapies, irrespective of whether they are a cause of aging, is of course, you know, for situations such as, you know, there may have been severe damage, injury, or trauma to an injury site, whereby upregulation of telomerase could potentially help, you know, the wound healing process. Things like that, you know, or where you've got a fibrotic disease like pulmonary fibrosis, which we talked about last time, certain types of anemia. You know, there are certain sort of spot applications that I think telomerase could be useful for anyway. So it's handy to have that in the toolkit, you know, whatever the case is, whether it is a cause or a consequence of aging, either way. And I think we'll find out as, you know. Well, like I asked you a few days ago, I said, do you think we'll reach a consensus on what is aging? And you seem pretty confident.
Yeah, I still do. I don't think telomerase is the one thing. I would doubt. If I would make a prediction, I would say if you would somehow engineer all of your stem cells to have telomerase operating all the time at optimal efficiency, you would still age. You would get other damage accumulating. This is their main, you know, telomeres are just one part of, you know, what can go wrong when cells divide. And I'll stress dividing cells, right? So most of your cells are not dividing. So telomerase might have other functions, but if we just look at its main catalytic function, it shouldn't have any effect on your neurons, for example, right? Your neuronal health. So,
yeah, TERT definitely has other functions. It turned up in the if you remember from Steve's lecture, it actually turned up again in the methylation clock.
yeah.
TERT was significant. Its presence was important, but not in relation to telomeres. So, like many things in biology, a lot of stuff. There's a lot of redundancy. There's a lot of multi-function built into all sorts of things. So
A lot of proteins get repurposed into structural role and then a signaling role. So
yeah, the same with NAD. They thought that all it really did for a long time was sort of help transport things around to enable redox reactions. Boy, were they surprised when they found out it was far more involved with cell signaling and all sorts. That's something we should look at as well. The NAD plus biology at some point as well, and why it is important in aging and not just caloric restriction as some people might think.
well, this kind of duality of functional roles that's really the heart of biology, isn't it? I mean, it starts with, like I like to tell my students, it goes right down to the information carrying molecules. If you take ATP, which is your energy currency, right, adenosine triphosphate, it is also your primary information molecule, RNA. So right there, you have this split personality, this dual functionality, and so that's why it's difficult to sometimes assign a specific function to a protein. So it could have multiple roles depending on the context.
Yeah, I mean, you see that a lot. You see things like TGF beta as well as a classic example. Everybody sort of thinks it's a pro-aging cytokine, but not necessarily, because in small amounts, in proper amounts, it also facilitates wound healing and all sorts of things like that. So it's not just a case of completely stamping it out. And same with TNF alpha and all the other signaling molecules. It's all about balance and calibration, really, at the end of the day. And the body's an extremely complicated machine, isn't it? So you can definitely tell it wasn't designed by humans. Otherwise, it'd be a lot easier to work out, wouldn't it?
I guess.
But it's had a long time to evolve and develop, and it's just mind-boggling how it all sort of works. Really, you'd think it wouldn't, but it does. It's just, I don't know. I kind of find that
more or less.
Yeah, more or less. But I kind of find it quite fascinating that how such a jumble of effectively many random systems all sort of work in unison. It's quite interesting, really. But can we do a better job? Yeah, probably in the future. And and why not? Why shouldn't we?
Well, to that, I'll say I hope so.
Well, I hope so. Yes, but I honestly, I can see in the future a time when we, when we'll be able to take control of our genetic destiny somewhat, and maybe iron out some of the wrinkles that evolution hasn't quite fixed, and help those little mitochondrials get to the nucleus, the mitochondrial DNA.
Yes, we can all speculate, can't we?
Well, sometimes it's fun, isn't it? Even scientists are allowed to speculate as long as you make it clear it's speculation.
Of course.
So but we'll see. So yeah, epigenetics is a possible subject for next time. So let's have a look. Let's have a look. And if anybody in the audience would like us to have a look at a paper, do send your suggestions in, because we do take requests as long as they're polite ones, right, Oliver?
Sure. As long as they're polite.
As long as they're polite, yeah.
Okay, guys. Well, I guess we're signing off this week. And when's the next Journal Club? 4 weeks from now?
It'll be sort of. It's usually towards the end of the month, as is
End
our.
of April.
Yeah, as is our sort of our way. I'm just looking at the calendar now, and I've got a good feeling somewhere around about the 24th, 25th, something like that.
Sounds about right.
But yeah, we'll announce that on the usual channels. We will announce that on the website and on the Facebook page, etc., and also in our wonderful Discord community as well. We'll keep you informed, which is what we do. And a big thanks to, again, the Lifespan Heroes. Without you, we couldn't produce this show. So thanks for that. And, we will see you next time. And thanks for joining us.
Yes, thank you.
Thank you very much.